What's up, folks? I don't even know if anybody's in here or not, but here we are reviewing Unit 2 AP Biology. So yeah, if you're here, just ask questions, and we'll keep going until there's no more questions or till I get tired. So feel free to chime in anytime. Use the chat over there. I think I've got it in subscriber only mode. That is my sly ploy to get you to subscribe. Some of you are probably just in here like chilling. You're afraid to say anything, which is fine too. So I can just start generally reviewing things until I get a question, if that's good. I can do that. So yeah, I'll wait a few more minutes and see if anybody joins in to ask a question. I mean, I'm assuming that I can be heard. Everything looks good on my end. Oh, there we go. Membrane transport, facilitated diffusion. Miss those days, and they've confused me a lot. I can do that. Yeah, let's do that. So, actually, you know what? We're gonna do 2.9 since it has all of them. And so let's look at 2.9, which is just mechanisms of membrane transport. And it's got facilitated diffusion on it as well. And then we'll look at 2.8 if we need to do that. So actually, let me go here first so that we can look at this concept of here. So we can look at this concept. Concentration gradient. Okay, so a couple of things you need to assume is that you understand what a solute and solvent is. The solvent is usually water, in this case, most of the time. And so in this particular picture, the white space would be the solvent. The blue hexagons have been dissolved into the solvent, and so therefore they are the solute, right? And the concept of a concentration gradient, one of the things that students were confused on, a concentration gradient is a thing. It's not an action. It is a thing that exists. Just like my roof, the roof that is over my head right now has a grade to it. That is a thing that exists. It is not something that's currently happening. It exists. There's a gradient here, right? There's a high concentration up here. There's a low concentration up here. I need to make my cursor bigger. Just realized it's not very big. So high concentration up here, low concentration up here. So there's currently a gradient, right? And in this particular instance, these molecules are traveling from a high concentration to a low concentration until there's no longer a gradient, right? And so you need to understand that concept, concentration gradient. Sometimes things will go with the gradient, and that means like, with means like, what kind of arrow is that? That direction, right? And then sometimes things will go against the gradient where they're going like low to high, right? And so we'll use that language as we talk about it. Pretty important. Two kinds of transport, passive and active transport. Passive transport is no energy. So think passive, that doesn't require any energy, and active requires energy, right? So now we'll go to here. Diffusion is the movement of, it's passive transport for one, it requires no energy, and it is the movement of materials from a high concentration to a low concentration. Great example of diffusion right here, right? So you get this dude dropping in this purple dye into this water, and it starts at an area of high concentration. The water around it is a low concentration, and eventually it is going to diffuse out until it is an equal concentration throughout the entire solution. And that is when it's called equilibrium. Equilibrium The technical definition of equilibrium is that there is no net movement of particles, meaning that things are moving equally. So another way of thinking about that. Simple diffusion. So if I go back up here to this, here's diffusion also, right? This time it's going across a membrane. It doesn't really matter. High concentration to low concentration. Most of the time when you see it, you're going to see it going across a membrane, right? So this is passive. No energy is required. Back to 2.9. Let's look at facilitated diffusion. So key word here is, so we know diffusion, right? What's facilitated mean? Facilitated is like helping something. So like if I facilitate something, that means I am assisting it or helping it. So facilitated diffusion is diffusion, so it's still high to low, passive transport, but it is using a protein channel. So this particular protein channel right here, wow, that's a bad color. Everything's just so, there, it's better. Protein channel here. And so they're going from high to low, right? They just have to use the channel. Why do they have to use the channel? Because they can't get across the membrane. They're probably polar, or they're probably charged or something along those lines. And so they can't make it across the membrane. And so they need this hot dog bun right here, which is a protein channel, to get from a high to a low. This is just another type of protein here. And we'll talk more about that particular kind of protein later. So that's facilitated diffusion. Osmosis. What is osmosis? Osmosis is just the fusion of water. But when you're dealing with osmosis, you're always talking about diffusion of water across a semi-permeable membrane. And there's a lot more going on with osmosis, right? Which if you were there for 2.8, you definitely saw that. So let's look here. So in this case, you've got water, which is the blue stuff, and you've got purple dots, which are purple dots. And over here you have a low concentration of purple, high concentration of purple. And so if you, and notice they're equal, right? Equal volume. And so if there's low purple, there's higher water here than here. And if this membrane prevents the movement of purple dots, then what is water going to do? Water is going to go from an area of high concentration to low concentration. Water moves just like anything else. It goes high to low. And so it moves and it creates this system like this until what? Until the water is equal concentration on both sides, or at equilibrium. So that is osmosis in the simple form. We'll get into tonicity, I'm sure. Can't imagine a situation where I wouldn't be asked questions about tonicity. And active transport. Active transport is using energy. There's ATP, our friend. Energy currency of the cell right there, causing this to happen. Why does it require energy? Because it's going from an area of low concentration. Oh, there's an arrow right there. To high concentration. It's pushing it up the hill. It is rolling a ball up the roof. However you want to look at it. It's going from low to high, and so it requires a push. And sometimes these proteins like this is like just a special protein for active transport. And then we talked about large molecule transport. We mentioned this a little bit the other day when we were talking about endosymbiosis, right? And how does a cell take in like a giant bean like this rather than like normal purple dots and blue dots? Well, if it wants to eat this bean, it can't. The bean can't diffuse across the membrane, and there's no proteins, right, to carry it, and so it has to make this like bubble, and so it like kind of reaches up and just pushes it, pulls it in, right, and it like pinches off a piece of itself, and like you could call this vesicle too. There's probably some technical like terminology. Which they said phagosome and vesicle here. Whatever. For our purposes, it's all fine. This temporary storage unit is going to be a vesicle, probably some lysosome or something that's going to come in and eat it. And so this is endocytosis, right? Why endo? Because it's bringing into, right? And then cytosis is just into the cell, right? Large particle into the cell. It could be phagocytosis, which is solid materials. It could be pinocytosis, which is liquids. It could be receptor-mediated endocytosis, which is just endocytosis, except for it needs a receptor. It can't just anything, right? Because the bean just didn't have a place to land, but these stars have a place to land. It's those little receptors, and that receptor being triggered is going to cause that too. So, yeah. Definitely will talk about solute potential. And then the opposite of endocytosis was exocytosis, which is here, right? Which is pushing large molecules out of the cell. Yeah, it could store them in a vacuole. It could immediately break them down. It could do any number of things. Oh boy. The AP classroom question. If you asked a specific thing, I could, but I definitely can't display that question on my screen because I would get in trouble for that. So, but if you have a specific thing, I could probably do that. But if I put it on my screen, I would definitely get in trouble. Just because they're not supposed to go out, even though they are. So yeah. Or you can come in first thing tomorrow morning and we can go over it too. Then we can do that for sure. So yeah, is that it on any questions on endo, exo, any of that stuff that you asked? And then I'll answer this question about solute potential. All right, then. Let's look at solute potential. Actually, you know what? I'm just going to use this picture right here. All right. So this is osmosis, right? And osmosis is water moving. Yeah, confidential information may not be distributed. Yeah, absolutely. It may not distribute it, even though it has been distributed to the ends of the earth. I am not going to contribute to that distribution. So we know that water, in this case, water has a high concentration here on the left, a lower concentration on the right, right? And we also know that if the words that we can use associated with this are, this one over here is hypertonic. Golly, it's just going to take me ever to write this out. Hyper. And this is hypo. Right? Okay. And water moves from hypo to hyper, right? We know that. And so what is causing that water to move? Right? What is causing the water to move? It's the fact that there's more solute over here. Right? And so the presence of solute is creating the potential for water to move. Right? It is creating the potential for water to move because water moves from hypo to hyper. So keep that in mind, right? So when we talk about solute potential, right? When we talk about solute potential, well, first of all, let's go back up here to this water potential. Water potential is pressure and solute, right? And pressure and solute are going to work to cause water to move. In this particular instance, pressure is 0, right? You could pretend pressure is 0, and then solute is a number, and those are working together to cause water to move. If there was some pressure like pushing down on this, then the pressure plus the solute would be working together to cause water to move. And that working together is called the total water potential, pressure plus solute, right? Pressure can be a positive or negative number because it can be positive pressure, it could be pushing down, or it could be negative pressure, it could be pulling. right? Solute is always going to be negative unless it is 0. Why is it always going to be negative? Because water moves from a high water potential to a low water potential. And so if you go back to this picture here, this is going to have a lower water potential. Let's say it's -4. And this one's going to have a water potential of -2 because there's some solute here. And so water moves from left to right because water moves from a high water potential, -2, to a low water potential, -4. And so when it comes to solute potential, there are a couple of awesome, George. Thanks for the kind words, man. Appreciate that. Awesome to hear. Congrats on the 5, by the way. Hope you're doing well, whatever your endeavors are. You do not need to memorize the equation for water potential. I will provide you with that number, or with that equation. So solute potential here. Notice the negative here. It's always going to be there. This number is negative unless it's 0. And you're going to multiply these numbers together. All these things will be given except for temperature. Temperature is going to be, it'll probably give it to you in Celsius, right? And so you're going to add 273 to it. Molar concentration will always be given. You're not going to have to solve for that. R is always 0.831. Another thing that's given. Literally all of this is on the formula sheet. Every single bit of it is. And the only thing you have to know is the ionization constant, which again, how many particles it makes in water. And we came up with this rule. Sugars, things that end in ose, are going to be 1. Everything else, acids, bases, salts, are 2. I don't know what an acid is. Is it a sugar? If yes, 1. If no, 2. That's just the way you can think about it that way. So if you see something like, this, and this is called potassium hydroxide, and you're like, I don't know what that is. Does it end in ose? No. So it's 2, right? And so that's the way you need to think about it. I love how it just now chose the, you know what? Maybe you misspelled this. Literally, that's been up there forever. So yeah, and this is going to give you a negative number, and that negative number is going to decide the value here, right? I came up with these arbitrarily, but there's going to decide some value, and that value is going to determine the solute potential. And then both sides are the total water potential. Both sides are going to have a number, right? This side has a water potential. This side has a water potential. You compare them to each other. Water moves from high to low. Good question. Other questions? Any other questions? That's great to hear, man. I love those kinds of stories. George, I don't know if you're still in here. Where are you from, if you don't mind? I'm just curious. I see that there are several people in here, which is good. But I don't know what you guys need to review. Honestly, this material is not too bad, right? It's not too bad. Until I get a question, I'll talk about cell size because it's important. So let's look at cell size. There's a question. Lipid bilayer, yep. There's a bilayer right here. All right. Actually, you know what? There we go. All right. So the lipid bilayer is made up of these little individual things called phospholipids. Here's a phospholipid. Phospholipid is amphipathic. What does that mean? it means that it is both a hydrophobic and hydrophilic. I should grow my beard out. No, it gets itchy. It gets pretty itchy. Hydrophobic and hydrophilic. It has a hydrophilic head. What does that mean? That means it likes water. What's another word that we could use for hydrophilic? We could say polar, right? We could say it's a polar head. What makes it polar? It's this phosphate. You don't need to know that. And then it's got a hydrophobic tails. So the hydrophobic means non-polar, doesn't like water, right? And that's going to cause it to do some weird stuff. You put a bunch of these phospholipids in some water, and they're going to organize themselves like this. Whereas the polar ends, the heads, are facing toward the water, right? The water's in the inside and the outside of the cell, in this case. And the hydrophobic portions, the tails, are facing toward each other, which kind of creates this membrane that doesn't allow things to pass. What is that? It looks like somebody drew that. Ooh, okay. So yeah, you get the idea, right? And so the bilayer is called a bilayer because it's made of two layers of phospholipids, and they're facing each other, whereas the polar regions are facing out and the non-polar regions are facing in. And there are some other things going on in this lipid bilayer, like these proteins here. And the proteins are also amphipathic. Why do they have to? Well, some of them are. Why does this have to be amphipathic? Well, because some of it needs to interact with the polar regions and some of it needs to interact with the non-polar regions. That's what that drawing was for. Because I probably drew that just like that, and I thought this was non-polar. And yeah, that's exactly what I did. Okay. And so the ones that go all the way through the membrane are called integral proteins. They go all the way through. And the ones, plasmodesmata is like just a vocab word. It's like the hallway between mine and Krug's room, but for cell walls. So, that's all it really is. Just a vocab thing. You can think of the reasons why plants might want them, to talk to each other, plant cells. So yeah, peripheral proteins are on the outside, inside of the cell membrane or the outside of the cell membrane. Bean is a protein, right? Yeah, the bean. Yeah, I probably should use more precise language. This bean is a protein. They always draw them like beans. That's actually why I like this picture, because it kind of shows you that whole primary, secondary, tertiary thing, right? That shows you more of a protein sort of structure as opposed to just, like weird beans stuck in the thing. So here's some more. These aren't really beans, but yeah, they're proteins. And those membrane proteins have several functions, which we talked about. And in this picture was just the idea that it's a fluid, it's always moving, it's a mosaic, it's composed of lots of things. And then we talked about cholesterol, which is a type of steroid that's found in the membrane, and we talked about glycolipids and glycoproteins, and we remember glyco means sugar, and so it's like a protein with a sugar on it, or a lipid with a sugar on it. That's all it is. Could very briefly go over 2 points. Yeah, I can go over that cell size for show. Yeah, let's do that. Okay, so let's talk about cell size. So big idea, cell size. Cells with a higher surface area to volume ratio are more efficient. Why is that? Well, why is that? So let's just look at these two cells right here. So here we have a cell that has a side of 1, right? So it has a surface area of 6. Why a surface area of 6? Because each side is a 1 by 1, right? So it's 1, and there's 6 sides, 1 by 1 by 6. 6 has a volume of 1, right? 1 times 1 times 1, length times width times height. So a surface area to volume ratio of 6 to 1, or just 6. So notice what happens here. The area, or the side, only increased by 5, right? And so the area is going to be a squared function, so the area is going to be 25, and the volume is going to be, so the area of this is going to be 25 times 6 is 150 right? Yeah, 150. All right, and the volume is going to be 5 times 5 times 5, which is 125. And so notice what happened here. When you increased the side by times 5, the volume increased by a factor of like, what, 25? Yeah, that's exactly right. And the area increased by a factor of 25, and the volume increased by a factor of 125. It's because volume increases faster than the surface area, meaning that the inside is going to increase faster than the area of the cell can keep up with. We saw this in our cube lab, right? Because the small cube was able to diffuse all the way through, whereas the big cubes weren't even close, right? And so it would be more efficient then to have a bunch of these little cells, right? 125 little cells, rather than having one big one. It's kind of like the Sonic ice versus the big 5-pound block of ice, right? And so it is more efficient to have a higher surface area to volume ratio than a lower. And it's literally just a comparison. Like if I were to ask you which cell, A or B, is more efficient, you could just look at this number and tell A is more efficient. Now, the way that the questions are going to be worded is it'll be some sort of sorcery, you know. It'll be like, which of these is least efficient? And then you're coming to it thinking, oh, what's the highest number? Well, it's not, because it asked least efficient, right? So this would be the big cell. And so that's the thing to watch out for. It's just the wording, which is literally everything we do. I don't know what that was, but it's gone. All right. And so, so be careful on the wording with that. Now, how is this going to play out application-wise? Well, I wrote some stuff. I wrote that. Okay. Application-wise, living things are always looking for ways to maximize surface area, right? Or use surface area to their advantage. Like creating lots of root hairs, or creating lots of surface area in the small intestine, or animals using that in order to cool off, because remember, elephants have tons of cells, and so they increase their surface area because their volume is too big, whereas mice don't have as many cells, and they lose heat really quickly, and so they decrease their surface area to volume ratio by balling up like this, right, to stay warm. And then we talked about not plasmodesmata. We talked about stomata as well to do that. So big idea, maintain a high surface area to volume ratio to maintain efficiency. That's the big idea there. Other questions? Anything else? Good questions. Good questions. If I was asked to describe the important organelles, plant, animal, in the fewest words possible. Okay, yeah, all right. So you mean like each one? Yeah, I can go over that 2.8 again for sure. Okay. So like ribosome in the least words possible. Ribosome is side of, or do you want me to like all of them together? It doesn't matter. I mean, ribosome in the least words possible is site of protein synthesis. ER, you'd have to talk about the smooth and rough. Yep, you're welcome. Smooth and rough ER, right? And the difference. And so the rough ER contains ribosomes, and since it contains ribosomes, it's going to be directly involved in protein synthesis. Smooth ER is detoxification, lipid synthesis, and so the rough ER, just a little bit more on that. Proteins are made here in the rough ER. They're going to be shipped to the Golgi, right, via this transport vesicle. I like how it's an incoming vesicle, like it's getting ready to land or something, right? And the Golgi is going to be a protein modification. How does it do that? What is it modifying? Well, it's adding things like glycos. It's adding sugars to proteins, right? That's exactly what it's doing. And it's going to do that, and then it's going to send it on its way. And where's it going to go? It's going to leave the cell, and it's going out of the cell via exocytosis. Notice how this is tied together. It's just wonderful. All tied together. All right. Mitochondria is the site of cellular respiration. And what is that? It is making ATP from breaking down glucose. And so it's very important. And lysosomes are digest things, basically break down things. Remember the prefix lys. Vacuoles are storage, temporary storage, or it could be like permanent. Usually it is permanent, like a plant's going to have a giant vacuole in it. Chloroplast site of photosynthesis. What else is there? Chloroplast, mitochondria. Just remember with these things we talked about a few parts, but nothing crazy. There's 2 reactions in the chloroplast, but we didn't really go into details of that. We talked about the thylakoid and the site of the light reactions and the Calvin cycles and the stroma. I think we mentioned those words, stroma, and I think so. We'll definitely talk about it next unit. Mitochondria remember chloroplasts and mitochondria have ribosomes, DNA, double membrane, all that stuff. Yeah. And so when it comes to organelles, keep it really simple and make sure you understand all the ones that we talked about have a, oh yeah, there's some more stuff on here. This was that whole ER plus Golgi, proteins from the cell. We talked about that. All of these are membranes. Oh, there it is. Oh, there's all the things. Okay, all these things have membranes to them, right? These lysosomes. The only organelle that we talked about that does not have a membrane is a ribosome, because a ribosome is more of like a cellular like particle as opposed to a membrane-bound organelle. Because remember, prokaryotes don't have any membrane-bound organelles. They do have ribosomes, but they don't have any of this other fun stuff, right? Only eukaryotic cells have that sort of thing. All right, so let's talk about tonicity again. Osmosis, diffusion of water. And so big idea with this. So it's going to ask you, like, you get a solution like this, and you have to decide which side has more solute in it. The one on the right has more solute, right? And so it's hypertonic. Gosh, should have left these up there. And this one's going to be then hypo. Remember we said how these words need each other, right? They're words of comparison, and so they need each other. And why are we using these words again? Hypertonic is more solute. Hypotonic is less solute. Water moves from hypotonic to hypertonic, right? Until both sides are at equal concentration. And we could say water as at equilibrium because the solute is isotonic, right? There's an equal concentration of solute on both sides, so there's going to be an equal movement of water. And tonicity. All right, so we talked about the plant cells. There's a potato over there. And we talked about how plant cells want to be, the inside of a plant cell wants to be hypertonic, right? Why does it want to be hypertonic? Because it wants water to come in, because it wants the outside to be hypo, so water can come in, so it can maintain this turgidity, so that it can hold its leaves up to the sky and do photosynthesis. Whereas an animal cell wants to be isotonic, right? It doesn't want to be hypo or hyper because it wants an equal movement of water in and out, because it doesn't want to blow up or it doesn't want to shrivel. And we came up with another way. We didn't come up with it at all. Somebody already did. We just learned about it. We talked about another way how water moves, and it's this idea of water potential. And water potential is the same. This is all the same. It's all the same. Water moves from hypo to hyper. Water also moves from high water potential to low water potential. And so you could say that this is high and this is low, right? Because it's the same thing. This is low water potential. Why? Probably because of all the solute. Why is the solute always low? Well, pressure, don't ignore that. Pressure is going to be positive or negative. It's given. It's always going to be given in the problem. Solute potential you may have to solve for. There's not going to be any calculations on the exam, but that doesn't mean that you won't have to use these numbers. You may be given a solute potential or something like that and have to use it. And here's how you solve solute potential. Ionization constant, this is the only thing you have to know. And remember, we said sugars are 1, acid bases are 2, and temperature you have to convert to Kelvin. And remember, this number is always negative, and so that's going to bring down. That's why I put that negative there. It's going to bring down the water potential, and water is going to want to move from a high water potential to low water potential. And so something like this, because this side's got so much solute, this might be like a -8, and this might be like a -1. And so water's going to move from high to low. That was the crash course on tonicity. I don't know if hopefully that was helpful. Anything else that you all have? Honestly, this is not bad. As I'm sitting here going over it, the concepts are not. This 2.8 was probably the trickiest part of it, and maybe a little bit of the cell size stuff, and it's not really that tricky. Yeah, you're welcome. But honestly, this stuff is not that bad. So I feel like you guys can do well. Format of the test, good question. It's 15 multiple choice and 1 FRQ. And I'm glad I need to put this in Google Classroom, but I can tell you all who are here. So we have a pep rally tomorrow afternoon, and they are cutting 10 minutes off of the end of 5th period. And so what I'm going to allow you all to do, if you want to come in at the end of lunch, you can come in and start your test at any point. I don't want you to, and I know some of you may have a class that you're in, and that obviously prohibits you. And I will give you time if you need it to finish. Obviously not way too much time, but I don't think it's going to be a problem. I don't think it's going to be a problem. But if you all need extra time, I want to make sure that you have it. So, but I don't think it will be an issue. This is not a difficult test. So, if you study, if you studied and you're prepared and you understand the concepts, it's not going to be that bad. Others. And some of you probably have chemistry 6th, and I know you guys have a test in there too. Yeah. Apoptosis, yes. Where we talked about apoptosis, programmed cell death. I don't remember if we talked about it. We have talked about it before, but yeah, let's talk about apoptosis. Apoptosis is programmed cell death. We did talk about it, and we talked about it with things like here, lysosomes. There it is, right there. It's like in black and white. All right. So, why might it happen? A cell either has a particular life cycle, and that life cycle's come to an end. A great example would be like the cells that used to exist between your fingers when you were developing. You used to have paddles, and then when you came time to get fingers instead of paddles, the skin between your fingers just dissolved, and the cells were like, okay, we're done here, and they just did apoptosis. They died on purpose. Or a cell detects a problem, like some sort of genetic problem, and would undergo apoptosis. And apoptosis is basically where the lysosomes inside the cell just kind of break open and basically make the entire cell the inside of a stomach, which if you can imagine your stomach breaking open and filling the rest of your body, it would be rough. So yeah, that is. Yes, that is what compartmentalization is. And the lysosomes are a great example of this. So like, here's this cell, right? And this cell is like doing things that cells do. It's like going around and eating like coffee beans, right? Or something. Looks like a bowl of cereal. Like the last couple of bites of cereal that it takes you forever to get with your spoon. Like it looks like that. Well, the lysosome has a bunch of potato chip. All right. It's funny, like people can just get away with calling me names, but if I called you guys names, like, it just wouldn't be cool. All right. So lysosome is in a particular compartment, right? And what's going on inside the lysosome is different than what's going on outside the lysosome. Just like what's going on inside my stomach is different what's going on outside my stomach, thankfully. So yeah, that is it. That is all compartmentalization is that you think of it as classrooms or like different rooms in your house or whatever. You know, I currently have this sliding door behind me shut in order to compartmentalize my house. Sometimes it gets loud around here. Good questions. Good questions. Anything else you guys have? I'm just sitting here like thinking about this unit, and it's just not that bad. Next unit we've got all this stuff, and so it gets a little more spicy. So you know, so we have a less spicy unit this time around. It's kind of nice. Other questions for me? Hopefully this is helpful. I like doing this. So, and that was pretty cool. The kid coming in here earlier, like telling me he made a 5 because he watched my videos. That's like super cool. Give you guys a few more minutes. I'm sitting here thinking I can't come up with anything in my head that I haven't gone over that is just sticking out as, you know, maybe this endosymbiosis. Gonna be super deep. I mean, probably not both of those supers. And then some of them are like really shallow. Some of them are really shallow. And then some of them are like, you know, use this page to answer the next 3 questions or something like that. And so it's a little deeper. The depth of the question is going to just be the amount of reading. It is not, there's no inherent complexity. It's just having to kind of parse through the question to what is this actually asking me. And honestly, the only way you get good at that is practice and time. So the more questions you're exposed to over the course of the year, the better you will get at it. And then that's why I preach you guys doing those practice questions so much, because it is so good to do the most of them you can. You cannot do too many practice questions. Nope, don't mind at all. Endosymbiosis. It's this idea of compartmentalization. And again, this definition is necessary to kind of understand what it is. So it's the idea that prokaryotic consumed another. They lived together, benefiting one another, created the first eukaryotic cells. Why eukaryotic cells? Again, let's get our definition. Has a nucleus, has membrane-bound organelles, right? So before that, there were prokaryotes, there weren't any membrane-bound organelles. It was just a cell with like some ribosomes. All right, so here we have this. Now this cell has a nucleus, but it doesn't matter. It doesn't really change our story any. right? And so this cell is just a prokaryotic cell, or this says eukaryotic cell, whatever, it doesn't matter. Just ignore the picture. This is a cell and it's chilling, right? And then it eats this, and this is an anaerobic cell, right? Anaerobic means no oxygen, right? And so it eats this aerobic cell, and the aerobic cell is able to use oxygen to do the process that we looked at earlier. This process, right? It's able to do cellular respiration, break down glucose, make ATP. And this cell wasn't able to do that before. And so something happens, right? We're not sure what happens. And rather than this cell with the purple eye, like consuming this little Cheeto cell, rather than consuming it and breaking it down for minerals like it would digest anything, they just live together, right? And so this is the whole word. If you look at this word, endosymbiosis, together, life inside, right? And so that's exactly what's happening. Inside, living together. And the idea is that these were the precursors to the first mitochondria. All right, so this would be like an animal cell, fungi, something like that. Well, take our first cell over here, the first mitochondria, and it eats a little green thing, right? Well, this little green thing is photosynthetic, meaning it's able to do photosynthesis, which is use the sun's energy to make glucose. Well, and this thing's able to use glucose to make ATP, so it would be nice if we had a little glucose making machine inside. Well, that's exactly what happens. So again, we don't know what happens, but something occurs to cause this chloroplast or this green thing not to be consumed, but for them to strike up a deal. And it's not what's, you know what I mean. And they live together, and these are the first chloroplasts, right? And so this would be like a plant cell or a precursor to a plant cell, because it's got mitochondria and chloroplasts. How do we know? Well, there's evidence. There's actually 4 bits of evidence. Mitochondria and chloroplasts both have their own DNA. I could exactly explain what amphipathic means. Give me just 1 second. All right, so they both have their DNA. They both have their own ribosomes. They both have a double membrane, an inner and an outer membrane, right? And this isn't on here. They both reproduce like a prokaryotic cell. So they reproduce independent of the rest of the cell, and they kind of just split like this. You don't need to know the name of it, but it's called binary fission. But you don't need to know that term. So that's evidence that this happened, right? Is that they demonstrate that they were probably at one time their own organisms. Hopefully that answers your question. If not, you can ask a follow-up to that. So let's look at amphipathic again real quick. Amphipathic plasma membrane. Amphipathic is the idea that it has both a hydrophobic and hydrophilic portion. Wow, I forgot that it does. What did I just do? Why did it earlier? It did the straight thing, whatever. So hydrophobic is water-fearing, right? And this would be like a non-polar portion, which is the tail. And hydrophilic, which is a polar portion, is the head. And it's just anything that has both. That's what that prefix amphi means, is both. Pathic. I don't know what pathic means. Any other questions? You're welcome. Glad to do it. Welcome. Welcome. All right. I will wait a few more minutes here, see if y'all have got anything else. But otherwise, it is about 9 o'clock. Tomorrow's Friday. got 6 days till fall break. It's kind of crazy. I can't hardly believe it. Can't believe it. Aquaporins. Aquaporins are basically an aquaporin is a fancy protein channel that just does water. That's it. It just has a name. I thought I had aquaporin on here somewhere, but I guess I don't. Yeah, water and aquaporins. There it is. So yeah, it's just a protein channel that allows the passage of water. All right. Well, if you guys have questions, I'll be there probably 7:15 in the morning. You don't have to come that early, but I will be there that early. So if you have questions about, like you mentioned, the AP Classroom question or others, please come in and we could talk about it. And otherwise, I feel really good about this test. I really do. I feel like you guys are going to do well on it. I don't feel like it's that difficult. So, all right. Well, it's been fun. Don't stay up too late. I'm going to be done for the evening.